Final speaker in this session is Thomas Dan, who's from Cambridge again, Department of Genetics, who's going to be talking about the epigenome. Thank you.
Okay, thanks for the invitation. So I'm slightly different talk from me. Talking about the epigenome and in particular DNA methylation. So what is DNA methylation? Why am I interested in it? So it's one of a great many marks that are layered on top of the ACTG genome sequence, but all seem to go under this slightly fuzzy label of epigenetics, which really does just mean on top of the genome. Why do I study this one in particular? This point. Lasers, yes. So why do I study this one in particular? It's the majority of the things under the epigenetic heading have a degree of fuzz to them, in that they're not modifications to DNA, they're modifications to histones. You have to worry about chromatin structure, where the nucleosomes are. This is still a tough problem, and I notice we have a chip session later in the week, which I hope will be addressing this. In contrast, DNA methylation is wonderfully simple. So it's a covalent modification to DNA itself. The only major one we find in the mammalian genomes. There is now a slight exception to that, but let's just worry about this one. It always occurs, or almost always occurs, at CPG sites, so we know where in the genome to go looking for it, and it's heritable. CPG sites obviously are symmetrical in double-stranded DNA. If we draw double-stranded DNA with methylated sites in blue, unmethylated in yellow, that's a fairly standard colour code that I'll be sticking to through the talk. Then there's this enzyme DNMT1 will recognise the rare hemimethylated sites and copy the methylation state across by methylating the unmethylated sites. So if you have semi-conservative replication of the DNA genome, you can follow that up with DNMT1 and get semi-conservative replication of the methylome as well. And this makes it very attractive as a place to go looking for a cellular memory, because this is a way that information can be stably passed across cell divisions. So how do we go looking for this? And I think you probably guessed that the bottom line is going to be sequencing, but there are several different platforms. They basically fall into two camps. Ones that look at individual methylatable sites, and these all rely on treating DNA with sodium bisulfide, which means that unmethylated cytosines read as thymidines instead. Alternative approaches looking at density of methyl groups on DNA using either antibodies or restriction enzymes. Antibodies to enrich the methylated DNA. Restriction enzymes can be used to either to deplete for unmethylated DNA in general. Either of these approaches you can assay using either microarrays or sequencing platforms, and I'm going to shamelessly switch between platforms depending on what's convenient, although the way everything is going is very much sequencing now. So what we've been using primarily is the MEDIP platform. This should be very familiar in that the experiment format is basically the same as CHIP, except in this case the antibody is recognising single-stranded DNA containing methylcytosine. So you've enriched for the methylated fraction of the genome. You either put it on a 2-channel microarray, or or sequence your enriched fragments. It would be nice if the analysis was basically the same as CHIP as well, and up to a point it is. You can take your sequence tags or whatever, align to the genome, stack them up, see peaks. Are the peaks the interesting bits? Probably not, or at least that's oversimplifying drastically. I'll show you just why. CpG dinucleotides, the sites for methylation, are not uniform across the genome. There's about 10-fold variation, and the tips of the highest peaks are called CpG islands, but it's not a binary system. That you don't either have normal genome or CpG island, you have a continuum between them. And the amount of enrichment you get for your MEDIP DNA depends not just on methylation state, but on CpG density. And in fact, if we add on the true methylation status of this genome region, again on a yellow to blue scale, you see that this point where we're getting the highest peak is actually only faint green. That's only slightly methylated, yet it's still yielding the highest peak. So we really want to be able to do something about this. So here's a slightly different way of looking at the experiment. I'm now going to switch for a bit into microarray space, just because it's slightly easier to explain, but everything I say can be applied to sequencing data as well, and to be honest, it works better. So you've shared your genome, you've got a set of fragments. You've looked at some place in the genome, either with a microarray probe or by stacking sequence reads up there. You know there's a set of initial genomic fragments that could have aligned with that probe. So and you then have some sites where you actually want to assay the methylation status, methylated, unmethylated. This site, you know, is within the probe, therefore you know every DNA fragment that hybridizes to that probe contains that site. As you go further away, the influence drops off gradually. So, what does the data look like? Again, coloured according to actual truth, which was, in this case, was obtained on a bisulfide platform. Ignore the x-axis for just a moment, look at the y-axis, look at the methylated points, look at the unmethylated points, look at the overlap. So we can't use this. However, add on a measure of CpG density on the x-axis, and suddenly you're getting close to separability between the methylated and unmethylated points. And if you squint a bit and turn up the brightness on this, you can see that the green, partially methylated points do fall broadly between the blue and the yellow. So there's at least some hope of being able to get some quantitative information out of this as well. So I think I should skip this a little bit in the interest of time. So, long story short, we write down a Bayesian model saying signal at a given probe depends on methylation state of the nearby CpG sites, weighted by distance. And implicit in this is that if you have a bunch of sites close to the probe, the signal that you can get is higher than if there's only one or two, or if the only sites in the neighbourhood lie a long way from the probe. So we wrote a program to do this now a couple of years ago. Some of my wonderful collaborators told me I had to call this the Bayesian tool for methylation analysis, so I went with it. A few more details. We're using nested sampling to do the Bayesian inference, which has worked very well. It takes non-negligible computer time to do a whole genome, but completely achievable. And this is something you can download and play with if you have any DNA methylation data. But important question: does it work? So this is another way of looking at the depressing reality of unprocessed immediate data. So in this case, the ground truth methylation data from bisulfide sequencing is on the x-axis. You'll see that the majority, but not all, sites in the genome are either almost entirely unmethylated or almost entirely methylated. Intermediate ones are rarer, although quite interesting. And again, you can see the depressing truth that the signals you're seeing from the unmethylated probes substantially overlap the signals you're seeing from the methylated regions. So what can we do with this? This is microarray data, so we do what everyone does with it and smooth it out a bit. This does slightly, although not dramatically, increase the correlation coefficient between MEDIP and the truth, but it's still in the 0.5 range. So I've said all along that CpG density is important. Can we do something simple about that? Well, it doesn't come much more simpler than just counting CpGs, dividing each score by CpG density in the corresponding window. Does this work? No, it doesn't. In fact, it reduces the correlation coefficient. And the reason is because the CpG density varies so wildly, you end up boosting the noise in the low CpG regions to basically stratospheric levels. So this is not a good idea. You can probably see where this is going. If you want to handle your errors a bit more carefully, taking the Bayesian approach is going to help you. Does it in this case? Yes, it does. So it would be nice to just have a line down the middle. We don't quite get there, but we come pretty close. Correlation coefficients consistently well above 0.8. In fact, for sequencing data, we're usually looking in the high 0.8s. Just to say, yes, this does work on sequencing data, and I think you can see why things work slightly better if we, again, look at signal against CpG density. The methylated and the unmethylated points are now really perfectly separable for all practical purposes. So, what does this look like? Well, some of this data is out there on genome browsers like Ensemble. You can look around. We can get genome-wide results using these days one lane of Selecta Illumina sequencing. So it's something that we can do relatively inexpensively, and we're starting to ramp up to do this genome-wide on panels of samples. So where do we go with this next in a technical sense? So one thing I would like to just digress on very briefly is that we're sort of seeing the end of the transition from microarrays to sequencing platforms. It's very tempting to use similar analysis approaches, but I don't think this is always a great idea, even though it often does work reasonably well. And one reason for that is that for arrays, the amount of technical, hard to explain noise is really hard, is pretty high. For sequencing, with some caveats like the compositional biases that people have already been talking about this, I'd say the level of technical variation is lower. However, on microarrays, you're assaying really very large numbers of molecules for each probe. Whereas for sequencing, if you stack tags up on the genome, the depths, with a few exceptions like RNA-seq of ribosomal genes, you're often only looking at tens or at the very most hundreds of tags in a particular region. People who PCR up stuff and look at 10,000-fold coverage are the exception that proved the rule. So one consideration is that the amount of stochastic noise in this data is actually higher. Technical noise lower, stochastic noise higher. On the whole, that's a good thing. Stochastic noise is easier to model, but it's something that should be taken into account for analysis. The other thing that I think is worth thinking about is that for microarrays, the dynamic range that you've got available at a given site is not necessarily fabulous, but reasonable. So if you look at the pipeline, you've got 16-bit scanners, and they've generally got about 7 or 8 low bits of noise. But as long as you've got the gain set to something sensible, you've probably got 8 bits of signals. So 256-fold meaningful dynamic range for every probe. That's not wonderful, but it's not bad. For sequencing, dynamic ranges varies a lot depending on your tag depth. And if you're looking at an experiment where your tags are only stacking up 10 deep, your dynamic range is actually seriously limited, which isn't necessarily a bad thing, but it's something that needs to be watched for. In particular, for things like MEDIP, where you're looking at signals that are distributed very widely across the genome, increasing the efficiency of enrichment isn't necessarily a good thing, because you can very easily swamp your available dynamic range. And so although I've said that you can get nice methylation results using a relatively modest amount of sequencing, if you then boost the efficiency of your immunoprecipitation, you can suddenly switch into a mode where you just get hundreds of tags from the most CpG-dense methylated parts of the genome and lose coverage everywhere else. So think about dynamic range, think about matching it up. Again, I think I should skip some stuff in the interest of time. This is just very briefly to say that although MEDIP is not a single CpG resolution technology, it's fragmenting the genome and enriching fragments. It is actually possible to get quite nice resolution out of it if you're careful with it. Just to finish off, what is this mark doing? The classical literature tells us that DNA methylation is repressing transcription, and on average that's probably true, but frankly I don't believe a word of it. There are many proteins that interact with methylated DNA or prefer not to interact with methylated DNA. There's a complex readout, and we're only just starting to put this into the network of genome regulation. It's a couple of little studies we're doing to try and get a handle on this. Firstly, we're starting to look at DNA methylation across populations of individuals. So one study that was published earlier this year, actually again done on an array platform, although it would be interesting to do this genome-wide using a sequencing platform, is to look at correlation between DNA methylation, promoters of genes, and the age of the individual who did this. And this is, I think, this is one of the first studies where people have actually looked at an epigenetic mark, not just in a small number of samples, but in well over 100 human samples. And long story short, yes, we do find many loci where DNA methylation tends to increase with age, rather fewer where it decreases, interestingly. So this is something that seems to be, at least at promoters, is being gained. The places that are gaining it are developmentally regulated genes. Interestingly, these are not genes that get downregulated with age. If anything, these tend to be genes where the expression level increases with age, although the correlation is quite weak. So this is one of the reasons why I think looking at DNA methylation, or in fact any of the other, most of the other epigenetic marks, as simple repressors or enhancers is probably not a great idea. I think it's an open question whether this is something that's causing dysregulation of the gene, or whether it's something that's a consequence of the dysregulation of the gene, or possibly even the cell's attempt to buffer against other mechanisms that are dysregulating the genes. But it's an interesting thing to follow up. We're also trying to, hopefully you've seen some of the pictures earlier on, the mosaic structure of methylated and unmethylated domains across the genome. If you zoom in, that structure remains intact until quite high resolutions, and in many cases the boundaries between methylated and unmethylated portions of the genome are quite sharp. So a student in my group went looking for what's going on actually at the boundaries, and found several sequence motifs, including this one, which, as far as we know, doesn't match any of the familiar set of transcription factors, although that's not saying much because the transcription factor motif databases aren't complete. This thing often occurs within about 20 bases of somewhere where you're seeing a sharp transition between a string of methylated CpGs and a string of unmethylated CpGs. So it's important to remember that although this is a pattern that's heritable across cell divisions, the whole thing pretty much gets wiped out in the germline and reinstated. The cell must be able to reimpose correct patterning, and this is presumably one of the things that generates that pattern. So I'd like to thank a number of wonderful collaborators who I've worked on. I should say, as hopefully my introductory slide gave away, I'm very much the computationalist. So Vardman in particular has done the lab side of all the MEDIP experiments. Sergey and Claire in my little group at Gurdon, who've been working on the aging and the boundary projects respectively. I always like to thank the people who keep my computers spinning, because I really would be lost without them. And the Wellcome Trust and Juvenile Diabetes Research Foundation for funding. Thank you.
Time for some questions.
So I think you showed us that the biologically meaningful methylation is typically clustered. But am I right in thinking you're not using that information when you're actually sort of estimating methylation status from your
Okay, so in the model we do actually have some neighbourhood clustering in there, yes. But we put that in as a prior, essentially. And yeah, it's clustered on a scale of up to about 500. Autocorrelation decays over about 500 bases to give you an idea of the clustering.
And related to that, what is the biological mechanism that leads to clustering?
That is an interesting question. I think there are DNA methylation has been observed to spread when people have looked at small regions in populations of cells so many cell lines tend to become methylated, and if you sort of look across multiple passages, there's definitely evidence that sometimes that you get small de novo methylated domains that gradually spread. So there is some sort of feedback loop between things that bind to methylated DNA and things that put extra methyl groups on. But I think the exact components of that loop haven't been pinned down yet, and how important that is in normal cellular function, again, I'm not sure has been pinned down.
Any other questions?
So as I understand it, you're able to make sort of inference about regions of
Yeah.
methylation, but you can't really zoom in. I guess if you're doing it by sequencing or indeed by microarray, your resolution's limited by the
Yeah.
lengths of the fragments that you're...
Well, the fragment length isn't necessarily the limiting effect, because you can to some extent deconvolve that out so long as you know the length distribution of your fragments, which you can for sequencing at least you can get at either by careful library preparation or just by doing paired end sequencing. And in fact, in the dying, you say this limits the resolution of microarray experiment, but actually in the dying days of ChIP-seq, there were several nice papers doing deconvolution on chip array data. So David Gifford's joint binding convolution method, for instance, and actually the resolution they were getting is quite impressive, and you can genuinely get resolution that is substantially better than the spacing of the probes on your array, let alone the length of your fragments. So don't completely write off enrichment-based methods if you want good resolution. It's more complicated than it looks at first glance.
Okay, well, let's thank all of our speakers from this session.